Generative AI is revolutionizing vaccine development by enabling rapid antigen design, predicting viral evolution, and accelerating the process of creating and optimizing vaccines. Here's a detailed look at some key generative AI tools and methods in this domain. AlphaFold predicts protein structures from amino acid sequences with near-experimental accuracy. This is critical for understanding how antigens and epitopes fold and interact with immune system components. Applications in vaccine development, mapping the structure of viral proteins, such as spike proteins in coronaviruses, to identify immunogenic regions for vaccine design. Aiding in reverse vaccinology by predicting the structures of pathogen proteins without requiring experimental data. Notable achievements: successfully predicted the structures of over 200 million proteins in collaboration with EMBL-EBI, facilitating vaccine research globally. Evescape predicts how viruses might mutate in response to immune pressure or vaccines. It uses generative models trained on evolutionary information from viral sequences. Applications in vaccine development, anticipating future viral mutations to design vaccines with broader efficacy, identifying conserved regions less prone to mutations, ensuring vaccine longevity. Key use case: designing vaccines that remain effective against rapidly evolving pathogens like influenza and SARS-CoV-2. VaxFormer is a generative transformer model specifically designed for antigenicity prediction and vaccine design. It generates antigenic protein sequences tailored for immune system activation. Applications in vaccine development, designing antigens for mRNA vaccines by generating spike protein sequences with optimal immunogenicity, predicting and engineering vaccine candidates for emerging variants of viruses. Special features: antigenicity control, which ensures the generated proteins effectively trigger immune responses while minimizing adverse effects. ABOT uses generative models and differential equations to design antibodies from scratch. It predicts structures and sequences for antibodies with specific binding affinities. Applications in vaccine development include developing monoclonal antibodies that can be used as part of therapeutic vaccines and creating antibody scaffolds that can serve as templates for future vaccine development. Notable success includes generating high-affinity antibodies for diseases like COVID-19 and other viral infections. FrameDiff applies diffusion models to generate new protein structures with specified functions. It uses iterative noise reduction techniques to refine generated protein designs. Applications in vaccine development include designing novel adjuvants that enhance vaccine efficacy and engineering proteins with specific immune-modulating properties for therapeutic vaccines. Unique Advantage allows precise control over the functional and structural properties of the designed proteins. Similar to AlphaFold, RosettaFold predicts protein structures and interactions. It also offers tools for designing new proteins with desired properties. Applications in vaccine development include modeling protein-protein interactions, such as between antigens and antibodies, and designing next-generation vaccines based on complex antigen structures. Protein MPNN uses machine learning to generate new protein sequences optimized for a given three-dimensional structure. Applications in vaccine development include redesigning antigen proteins to improve stability and immunogenicity. Additionally, it involves creating sequence libraries for high-throughput screening in vaccine development pipelines. Generative AI assists in designing mRNA sequences for vaccines, optimizing codon usage, stability, and translation efficiency. This ensures the mRNA produces the desired antigen effectively. Key tools: AI-driven platforms for optimizing mRNA sequences for vaccines, like Pfizer-BioNTech's and Moderna's COVID-19 vaccines. Generative AI tools are rapidly transforming vaccine development, offering unprecedented speed, precision, and adaptability. These innovations are paving the way for more effective and accessible vaccines for global health challenges. Validation: synthesized antigens and antibodies need experimental validation to confirm efficacy and safety. Ethical concerns: ensuring that generative AI designs are used responsibly and equitably in vaccine production. Integration with experimental methods: combining AI predictions with high-throughput experimental validation to accelerate development timelines. Regulatory approval: adapting regulatory frameworks to accommodate vaccines developed with AI-driven processes. Generative AI tools are rapidly transforming vaccine development, offering unprecedented speed, precision, and adaptability. These innovations are paving the way for more effective and accessible vaccines for global health challenges.